Welcome to Side of Science.
With Stacey and Clinton, because every story has a side of science.
On today's episode, we are talking DNA family drama, your genetic neighbourhood, a 90s video game gene link, and answering the question, what is the DNA we share?
Clinton, I was researching recently and came across a fascinating story about a woman, Lydia Fairchild.
I love the name.
Yeah, well, it becomes a little bit ironic, actually, because she gave birth to children who didn't genetically match her blood. So they actually thought that she wasn't their mother.
That maybe it was like switched at birth.
Yeah, something like that. And so what's happened is this woman actually she's a chimera, she has two sets of DNA. And the DNA that her children have is not hers, but is her twins, essentially, that she and her twin fused at birth. So she has two sets, and she's not actually the mother of these children.
How does that happen?
Well, yeah, that's what I wondered. So I deep dived into this because how is it not a 50-50 split, and which parts are her and which part are her twin? And so...
and how does she just not have 4 arms and 2 heads? Like, she's got 2 sets of DNA.
She does, but it comes down to when they fused. And so this is like within days of fertilization. So it would have to be really rare. What's happened is her mum essentially has released 2 eggs at the same time. So they've both been fertilized, and that would have resulted in fraternal twins. But very early on, those 2 cells have fused together. And in that process, there's 3 layers essentially that start to form really early. So they are the ectoderm, which becomes the skin, hair, and the nervous system. The mesoderm, which is your muscle, bones, and blood. And then the endoderm, which is your gut, your lungs, and your digestive organs.
And what are those 3? Are they types of genes or types of...
They're tissue layers, essentially, that start to happen when the baby develops. And so there'll be an outer, a middle, and an inner. And depending on where that fusing happens, then the patterns of cell growth will determine what kind of organ or structure these cells become. So at fertilization, the cells can be anything they want to be. Isn't
magical.
That nice? Yeah. So depending on where they are is what organs they'll turn into. Like stem cells, we hear that they can kind of be put into a liver, there'll be a liver. Put it into an eye, there'll be an eye cell. So this is super early. And what seems to have happened with this particular case is that the second twin has not been the dominant cells. It's not taken on the hair and blood and nails. Instead, these particular twin B has become the gonads. So they've turned into her reproductive organs. So to actually get to find out that she was the mother, they took cells from her uterus to find the match and then found out what had happened. She would have had no idea otherwise.
It's wild, right?
Yeah.
But it really does. Since you've told me this story, it's got me wondering really about this question of really what makes us unique. Like, what are the things about us that make us different to the person next to you? But more closely in this case, basically what would have been her twin sister.
Yeah.
Like, they're still different enough even in that situation that they're coming up as essentially different sets of DNA markers.
Yeah. So she's more like the auntie of these children than the actual mum. So the DNA discrepancy is larger than it would be from being maternal match. Yeah.
Which means if her kids don't agree with her, she can say, well, make sure you listen to your aunt then.
That's right, yeah.
It's very convenient.
Yeah, well, hopefully she feels like that, yeah.
So can we go back then to like just some 101s around DNA and how that works? I recall vaguely that it's basically you get the sets from your mother and father.
Mm-hmm. Yeah, so DNA, deoxyribonucleic acid, is the instruction manual for all living things. So every cell will have DNA. In simple terms, it's like a recipe book. It carries the instructions to make proteins, which do everything from building muscles to giving your eyes colour. So DNA is like a double helix twisted ladder. We don't really need to go into the chemistry of it, but it's really just made up of 4 repeating units, A, C, T, and G. And the way that they are arranged in that code is what makes us unique.
And I understand that we share a lot of the same DNA. Like it's like what I think I'd read it's like 99% of our DNA is shared between humans. It's actually only the very small percentage that is making us unique, essentially.
Yeah. And it's interesting because I guess it depends what part of the DNA you're looking at. Genes are sections of DNA. And these genes that code for these structural units, they need to be the certain recipe to have a certain outcome. So if you've got an enzyme that is involved in respiration, for example, it needs to have a set structure. And so the recipe for that would have to be the same in you, in me, in my cat, in my dog, in my dad, in my brother. Otherwise, it doesn't work. And then a fetus wouldn't be viable.
Is that when those structures are broken, for example, is that when you can get issues early on in like development?
Yeah, definitely. So conserved proteins, those proteins that are absolutely essential for life. So I'm not talking about eye colour, which obviously you can have different versions of the same gene. These particular conserved genes that are responsible for survival, if something is wrong with the genetic code that codes for those, then quite often the pregnancy wouldn't be viable.
So just to clarify then from what you're saying, essentially that 99% of shared DNA is it's really for all that essential stuff that basically makes us able to just be a being.
Yeah. Yeah, and so we will have changes that we get from our parents, like different versions of these genes, and also epigenetics, which are slight changes on top of the DNA, like a methylation, so basically a sugar or a protein on top, and that can switch on or off genes. And so that gives us variation within the gene, like height or weight, things like that. But essentially, all of the DNA for these genes is going to be very similar.
So then that 1%, is that what is giving us the more visible unique characteristics like hair colour, eye colour?
Yeah, good question. It can be. So the 1% difference or variation can be the alleles. So alleles are variations of the same gene. So if we've got a gene on the DNA that codes for eye colour, then you can obviously have different colours. And so those different colours are the alleles, the options that are available. The other differences can be found in regions of DNA that are non-coding. So they're not actually genes, or they don't code for a protein. And so these can be single base differences. So we spoke about the ACTG, where there might be a C in one person. Actually, in another person, it's a G. So there's a single base difference. Or within the DNA, we often have repeats of sections. So like a GAC might repeat 4 times, 15 times, or 20 times in a non-coding region or more. But those repeat differences, or we call them short tandem repeats, they're also points of difference.
Okay, can I do a quick check-in then to make sure I'm following?
Yes.
99% shared, 1% not. Mm-hmm. So that's part of what makes us unique. There is coding DNA.
Yep.
That is the DNA that essentially makes us who we are.
Yeah, that's what we're turning the DNA into a protein.
Non-coding DNA, on the other hand, is more, as I understand it, much more like operational structural DNA.
Yeah, so it doesn't make proteins, but it's not useless. It can actually just regulate genes, so turning them on and off, repeating elements or protecting the ends of chromosomes. So when a protein is made, it will be spliced and it'll get rid of the non-coding DNA and just code for the protein.
And then a couple of other key concepts we've covered as well are alleles, mm-hmm. which are...
They're the variations of the genes. So if we think about DNA, then it's broken into sections called genes, and they will code for a protein. And then the alleles are different versions of the same gene. So if you've got hair gene, then different colours are the different alleles.
And they can be across the full 100% range of the DNA, those alleles. Like we can get that uniqueness being expressed through them as well as in that 1%.
Yeah. So your changes can be through the alleles and also through epigenetics. So epigenetic changes are DNA methylation, which just means there's a CH3 group, methyl group, that's added to DNA, usually to a C base, and that can switch genes on or off, or it can be like a sticky note that tells the body, do not read this. And so you'll get variation within the whole range of the DNA there too.
Okay, now we've got an understanding of the structure of DNA. Mm-hmm. Let's go back to poor Lydia Fairchild and her 3 children. What would they have been looking for in her DNA to then match it to her children, or in the initial case, not matching?
Yeah, so as I was saying before, a lot of the coding DNA will need to be the same. So they actually look at the non-coding regions, and specifically for forensics or for matching maternity, paternity, they'll look at short tandem repeats. So these are repeating sequences in non-coding regions that help to profile different people because different numbers of repeats will be at certain locations. So if you think of a location like a street number, then you can check that particular street number for person A and compare it to person B, see how many repeats they have at that particular location, and the number of repeats, the more similar they are, the more closely related those two people will be. So in Australia, forensic labs typically use 13 to 15 different locations across the DNA to check for.
So they go basically to 13 houses and look and go, how many repeats are these?
Yep, at each house, exactly. In the USA, it's actually 20.
Oh, wow.
And so that's because the probability of a false match is less than 1 in a billion. So because they have a bigger population, they have to check more people. There's always a slightly higher chance 2 people will share more genetic traits just through chance. So this kind of stuff, if it's used in forensics and not just paternity, then you need to be statistically significant, and they usually use other evidence as well. Does that make sense?
Totally. So particularly in that case, because the US population is so much bigger, the chance that there could be some repeats at similar houses, at similar locations, is slightly high. So they need to basically check more houses.
Exactly. Yeah. So paternity testing or maternity testing, usually 15 to 20 are compared at each location, and the child should have 1 allele from mum and 1 from dad. And if the alleged mother or father doesn't match on 3 or more, then it's excluded. So you'd be more likely an uncle or an auntie. If the matches line up, then the probability is 99.99%.
And I'm guessing they're looking at the non-coding DNA because that's where there's going to be a bit more similarity, whereas in the coding DNA, it's more likely to produce that uniqueness.
Yeah, so these short tandem repeats are only in the non-coding regions, and the number of repeats that you get will be more closely related within families, or more similar, I should say, in families that are related. Whereas with using protein coding genes to identify people, they're just too identical in everybody because they're conserved. They have to be highly conserved. There won't be mutations in them because in a lot of genes, if there is a mutation, there can be a problem. Only in those really, like I was saying, those survival proteins, some mutations can be beneficial and will survive. Like blue eyes is a mutation.
I'm looking at your blue eyes. Yeah. It's a wonderful mutation.
Thanks.
So, Stacey, if I do one of those very questionable online ancestry tests, which I've never done, by the way, I'm fascinated to do one, are the scientists in the lab that I'm posting this off to looking along the same kind of streets for the same kind of repeats in the same kind of houses? Like, are we basically following the same process?
A similar process, but instead of looking at short tandem repeats, we're now looking for snips. So a SNP is a single nucleotide polymorphism. So poly means numerous and morphism just means a difference. And then the single nucleotide just means one of those CAGTs is different in a particular spot or house. So SNPs, these single nucleotides, they're more stable over long time periods. So a single base change at a specific spot, like an A to a G, doesn't change very often. The stability means it's preserved over thousands of years, helping scientists track those kind of ancient ancestral and migration patterns and populations. So we're zooming in on like a tiny genetic signature that sticks around, because those STRs, the short tandem repeats that we look for within familial comparisons, they change a lot. So with even from mum to child, because you inherit half of the DNA, that there'll be a lot more
variation happening.
Yeah.
Whereas what you're talking about is really a micro view, but actually influenced at a really macro level over time. So they're more like when major migrations happen over thousands of years, etc.
Yeah, exactly, because you'll either have that original base or a mutated one. And so then you can kind of standardize and compare across really large databases. And so one person, if we're looking at STRs, they vary by length, not just content. So it's a lot harder to do that. So when we're looking at ancestry, you can sort of look at these particular SNP coding regions on a chromosome and say that, okay, well, 90% of people in Northern Europe share this particular marker, whereas only 10% in East Asia. So that's where you get the ancestry informative markers that you get that information from.
Are there examples of these SNPs that are carrying through ancestors, through generations?
Yeah, so some of the visible ones that could come through, or physical changes or structural changes that people might know about. One of them is Viking Finger, it's called. And so this is a condition where the connective tissue in the palm thickens, causing the fingers, usually the ring finger or the pinky finger, to curl inward over time. So it's sometimes called Viking disease or Celtic hand. So most common in people of Northern European descent, especially Scandinavians, Irish and Scots. And it's thought to have a genetic component passed down from Viking or Celtic ancestors. And so there are multiple SNPs that have been linked to the condition, or the SNPs, though it's not caused by a single gene, but a 2011 study identified 9 SNPs associated with a higher risk.
Next time you're in an argument, you can just say you're giving someone the Viking finger, or that you have Viking finger.
It would look a bit different. I'm going to try that. Yeah.
Okay, just while we're on strange names like Viking Finger, which I do love, I did also come across in my research a family of genes that are in humans that are named after hedgehogs. And I just love that we have the Indian hedgehog gene, the desert hedgehog gene, and then the best one of all, the Sonic hedgehog.
So this is interesting. These are in humans too, and they are involved in embryonic development. So similar to what we were talking about before with the 2 twins essentially that fuse. And so this means that the reason that the face is in the middle of your head is down to a Sonic the Hedgehog protein.
Which is great, which apparently the story goes, as I saw online, is that there was a group of researchers that identified these in flies, and they named them hedgehog due to the fact that some mutations with these genes caused basically spikes,
right,
which hence the hedgehog, and that they had settled on a third name, not Sonic, I think a more traditional hedgehog name, but then one of the more junior researchers that night, it's in the 90s, they were a little bit more liberal with their naming of genes, evidently. He sees an ad for a new video game called Sonic the Hedgehog, and he loves Sonic Youth, and he just kind of goes, oh, that sounds fun. And
it does sound fun, but it makes it trivial, doesn't it? Because I think if this actually signalling protein is low, then your brain might not actually split into two hemispheres.
Yeah, and I had read it's related to all sorts of development early on, like your teeth, all these kind of core structures in the embryonic development.
I'm all for it. I think if you find a protein, you name it what you want. It makes science a lot more interesting too, doesn't it?
Well, there were actually some other really interesting ones, like the Ken and Barbie gene. There was a Beethoven gene. There's also Cookie Monster because of the shape of the cells that they were seeing under the microscope. It looked like kind of Cookie Monster's eye. So there's a bunch of other ones that people can go and research in their own time.
Yeah. Find your favourite.
This has got me thinking of one last thing, which is cloning. So when they clone us in the future to keep this podcast going for eternity, are they literally...
Have they inquired yet?
They're incoming.
Okay.
Are they literally taking a photocopy of all the DNA, and therefore then the question is, are you still unique then?
Good question. You will be unique. Yeah. So they are just taking a copy. So I'll tell you about, do you remember Dolly, the sheep?
Okay. Absolutely. It was my year 12 English assessment.
Was it really?
It really was.
Wow. Did it come up why they named her Dolly?
Because of, no, I can't recall.
Okay. So Dolly was essentially taken from a mammary cell, and Dolly Parton's known for her mammaries. So that was the link there. But they took the mammary cell and then they took an egg cell from a donor. They remove the DNA that's in the egg cell and they replace it with Dolly's cell. And then they they combine the egg, which now has no DNA. They put Dolly's DNA in there and then they combine those with an electric shock, which triggers it to develop, so to reproduce, divide. And then they place it into a surrogate ewe, female sheep, and that will give birth to essentially Dolly, which has the exact same DNA as the original, so it's a clone. But there will still be uniqueness. And so that's because of gene expression. So the epigenetics that I spoke about before, which can turn genes on or off, that can happen due to responses in the environment. So things like diet, stress, sleep, social experiences, smoking, like drug taking. I don't know if Dolly was a rager, but that can all have different impacts. So there has been people who've wanted to clone like their pets, but their pets won't necessarily look the same because of these kind of differences, even though the DNA is the same. And
we're talking about it at a cell dividing, then becoming an embryo, the embryo growing, that then becoming a live birth. Like there's a lot that can happen, I think is what you're saying in that process, that can influence the changes beyond just a photocopy of the genes. Yeah, so that's in development, but then even like a nature versus nurture argument, like if they wanted to clone us for the podcast, there's no guarantee that my clone would be as funny as I am because of the experiences that I've had and the way that I think. Makes sense, complete sense. Okay, so my year 12 assessment using Dolly the sheep was back in 1997. That's a long time. Why haven't we got clones yet? Yeah, so it sounds pretty simple. You copy the DNA and grow a new animal, but it's biologically messy and inefficient. So you are putting adult DNA into an egg and then trying to reset it back to an embryonic state. So it's a bit like trying to wipe a chalkboard with permanent marker. It's really hard to erase all the epigenetic tags. And so genes that may be turned on or off incorrectly during development will cause problems. So I think cloned embryos have problems with placenta formation, organ development, and immune system regulation. So a lot of them actually a high miscarriage rate because they fail to implant or die early, or the clones have shortened lifespans. I think Dolly the sheep had premature signs of aging, the poor girl. So, unlike the real Dolly Parton. Yes, who's preserved. So these new dire wolves that have been created, it's not quite Jurassic Park, but the actual dire wolves went extinct about 10,000 years ago. And so the DNA that they have is not easily
kind of transcribed. We don't know 100% what it was. So they've kind of had a best guess at it, and they have altered grey wolves to be, so I think, 20 genes have been edited, which is less editing than I do on a selfie. So they've kind of changed them a little bit to have more dire wolf characteristics. But these have been cloned in a little bit of a different way, so they're not having the same epigenetic reprogramming issues. And this is possibly why these puppies are now lasting longer, and hopefully we'll learn a lot from the process. In saying that, there are a lot of ethical issues in cloning and bringing back extinct species. We've all seen Jurassic Park. Is this a good idea?
Who knows? Time will tell, right?
Yeah.
So it brings us back to our question. What is the DNA that we share?
Yeah, so the DNA we share is going to resemble those who we're most closest to. The more related you are, the more similarities you'll have, unless, of course, you are your own child's auntie.
Which never happens unless you're Lydia Fairchild.
Right.
And how do we bring this to the classroom? So much to unpack, from Sonic Hedgehog to Dolly the sheep to Lydia Fairchild.
So proteins, protein structure and function, DNA, protein structure and function, profiling for forensics and ancestry, ethics, cloning.
The list goes on.
Yeah, a lot of biology. All the good stuff.